Hello, everybody. Welcome to this International Mountain Days webinar. We will try and go through the OmicsOffice ExpressionExplorer, which is a tool we provide that will help you to navigate through those mountains of data that are accumulating in the hard drives nowadays. I am David Gonzalez from Integromics. And I have been working on expression analysis for a few years, previously in RNA-seq, and now since a year I've been working for Integromics, and we've been working on expression analysis on different platforms, including RNA-seq, microarray, and qPCR. Okay, so let's start by having a look at the overview of what we will be seeing today. First, we will go through an outline of what we will be viewing today. So we will start with some of the current challenges present in the omics data analysis. We will speak a bit about the importance of the visualization in this type of analysis. And also, we'll go through an overview of OmicsOffice. After this, we will go into OmicsOffice and use the ExpressionExplorer, which is one of the elements of the OmicsOffice suite, in order to explore an analysis example using, in this case, some data from the Illumina Human BodyMap, which has been analyzed with a third-party software. And finally, we will speak a bit about what conclusions we can get on this type of data exploration. One thing I would like to mention is that if you have any question at any time, feel free to write it in the question box that you will have available in the GoToMeeting, and at the end of the webinar we will address as many questions as possible. Okay, so challenges in the omics data analysis used to be, until relatively recently, the data generation, which was a wet lab process, so usually it would take quite a while to get all the data, and then the analysis could be done in a relatively short period of time. However, with the increased use of high throughput technologies in different areas, this has now started to change. So thanks to this rapid adoption of these high-throughput technologies in the omics field, now one of the main bottlenecks in this type of projects is the analysis and the interpretation of the data that's generated, which is a process that is done usually in silico, so it's a dry lab process. And as an example, we can see that RT-qPCR can generate thousands of reactions per run. With microarray technology, you can go to tens of thousands, and with NGS, you can easily reach the terabytes of data. So what used to be a relatively short part of the project is increasingly becoming one of the steps that will take longer. If we look at the different steps that we would see in one of these analyses, once we have collected the raw files, this can be qPCR, microarray, or RNA-seq in the case of expression analysis, or ChIP-seq. We would have these raw data files. We would want to go through a quality control process in which we make sure that the data we have is correct, in order to make sure that any conclusions we might obtain are also valid. And after this, depending on what technology we are using, we would proceed to some type of normalization. Usually we are comparing different conditions, so we would go through a differential expression step with whatever statistical test we think are suitable. And finally, we would look at the data set in the context of the biology, usually through functional analysis, looking at genes that are enriched in certain pathways, looking at expression patterns that may be different in different tissues. In all these processes, frequently the importance of visualization is usually underestimated. So usually visualizations are considered as part of the final reporting and not part of the analysis. So in many cases, they are not even considered in the initial steps of the analysis. However, if you can represent the data in a correct manner, this can improve data exploration quite a lot and speed up the analysis in many cases, as it can help to identify properties of the data, be it elements such as outliers or patterns or trends in the differential expression. So how can these visualizations help in this analysis? So if we are looking at a complex data set, we can use as an example a big simplification. So for this case, we will use this small data set that was created last century by Francis Ensembl. This is a set of 4 different data sets, which have very similar statistical parameters. So if you look at most of the statistical parameters that are used in standard analysis, these data sets are pretty similar. So you have very similar medians, very similar average, and standard deviation, and the number of data points is the same. However, if we start exploring this data visually, we can already see with a very simple representation, such as a box plot, that we can distinguish already different patterns in the data. Although in some cases, you can see that the 2 last data sets still look pretty similar. If we continue using the visual exploration, now we can see that the data are widely different. And this is something that, just by looking at the numbers, would not have been so easy to detect. This, of course, is an extreme example. So you would probably easily see, looking at the data, these problems. But what I want to do is just make a point on this fact that by using visual exploration, you can easily detect by eye things that might be strange in the data, or large patterns that might be hidden if you only look at the standard parameters that are used frequently in the analysis. So if we summarize the way that we would perform an analysis, as I showed in the previous slide, we could divide the process in different steps. So we could have one step where we would look at the data quality, one step where we would interpret the data, that may include differential expression, functional analysis, and any other type of data analysis you want to perform, and the presentation of the data. In all of these, you can have an important influence by having the correct type of visualizations. For example, when looking at data quality, we want to see if the data is good enough to proceed, and if so, what would be the best way to proceed. And good visualizations can help decide this. You can easily see outliers, for example, or determine if data is distributed in a normal way or not, this type of, which will help in downstream analysis. When you want to interpret the data, when you're looking at functional relevance. You can also see by using visualizations such as clusterings or heat maps that if there is different trends or patterns in this data. And of course, what everybody uses visualizations for is the final data presentation. And in this step, the adequate visualizations aim to provide the maximum information with the minimum clutter in the screen. So in order to address these different points, we have been developing a solution that currently addresses the analysis of expression data. And this solution would be the OmicsOffice solution that we provide. So if we have an overview of what this OmicsOffice suite is, we can see that it has several parts. So it has a core element, which is which we call OmicsOffice Core, of course. And this contains a set of tools which can be used in order to analyze any type of data. So this data can come from any third party, such as you could be analyzing your data using solutions from Partec, from CLC, or non-commercial solutions such as R. And you can get a set of expression levels, and these can be loaded into OmicsOffice. This OmicsOffice is running on a business intelligence platform called Spotfire, which has been adopted by many types of industry. And by adding the OmicsOffice capabilities, what we are doing is bringing this business intelligence software solution to the world of biological analysis. And currently, this OmicsOffice solution is distributed exclusively by PerkinElmer. So if you are interested in it, you can contact us or them, and any of us will be happy to provide you with any additional information. In any case, here, once we load our third-party data into the OmicsOffice, we can use also the ExpressionExplorer, which will provide us with a set of visualizations and analysis in an automated way that will help in the exploration of the data. And the OmicsOffice tools can be used for any other type of analysis. We will go in more detail in the demo at the end of the presentation. In addition to this, if you don't have a different solution, we ourselves provide some automated workflows for the analysis of different types of data, such as qPCR, microarrays, and NGS, in this case, RNA-seq or ChIP-seq. So altogether, you could, we would provide this package, which would be the OmicsOffice Pro. However, today we will just focus on the OmicsOffice Core without any of the workflows. And we will focus on the use of the ExpressionExplorer on a data set that has been analyzed initially outside of OmicsOffice. So it comes from different software. In order to do this, we have selected data from the Human BodyMap, the Illumina Human BodyMap. And this data contains information from 16 different human tissues. These have been processed, they have been downloaded, aligned, and the expression levels have been calculated. So that what we have is basically a table that contains just the FPKM values for each of the different tissues in each of the different genes. Once we have this, we will start by loading this into OmicsOffice, and we will perform a set of comparisons between the some of the different tissues, and also we will perform a comparison between the reads that have been generated using 50 base pairs and 75 base pairs. So there's two different read lengths that have been used. So now that we have the data, we just load it into the OmicsOffice suite, and we can start asking questions. So the way we can ask these questions by using ExpressionExplorer is by using the interactivity that it provides us. So it provides us with many visualizations that are fully interactive and very intuitive to understand. It's easy to export data and figures. You have full freedom to rearrange these visualizations. And of course, we try to make it as user-friendly as possible, so it's very easy to upload the data and start analysis. So a few of the typical questions that can be asked would be are shown here. So for example, we can see if there's difference between the different technologies, so 50 base pairs and 75 base pairs. Are there similar expression profiles in different tissues? If there are significant differences between when we compare different tissues, which are the top expressed genes in the different tissues. If there are any functional categories that are more represented or less represented, or how similar the top expressed genes are in the different tissues. So I'll use this as an example. So the way we will do it is, in first place, we will just load the data. So in this case, it's the expression data in the form of a table of PKMs. In the first case, we will use the OmicsOffice tools, and we will generate differential expression information. So here what we are doing is we are loading the expression data, and we are looking at cases where there's genes that are differentially expressed between different tissues. We have done a set of comparisons. So in this case, we've done 5 comparisons between different tissues, and we've also compared the reads, the expression levels generated from different read links to see if there's a significant difference between the expressions calculated using 50 base pairs and 75 base pairs. And now that we have these two information, the expression data and the differential expression data, what we will do is load it into the ExpressionExplorer. Once we have loaded this information in the ExpressionExplorer, we will execute the ExpressionExplorer workflow, and then we will go through the document that's generated in order to see how we can explore these results. So now I will just pass over to the demo. Okay, so when we open Spotfire, which is where the OmicsOffice platform runs, we will be presented with this screen. So in order to explore the data, the first thing we do is we will open the ExpressionExplorer, create a new analysis, and the only thing we have to do is load the different data. So we will just load the expression data. So here you can see it's basically we have a set of genes as rows and a set of samples with the expression levels. We will add the expression for the different tissues. We will add an experimental design. This will indicate the program, as you can see here, what dataset belongs to what. So we have different datasets that have been run with different read lengths, and we also have a separate category tissue where we have the different datasets from the different tissues. We will also add information on the differential expression. So this, once again, is a table with gene information, fold change information, and significance information for each of the different comparisons. And here we can load each of the different comparisons. For example, in the tissue, we will compare for the comparison of adipose tissue versus adrenal tissue. We select adjusted p-value for adip versus adrenal, and we assign it. The same thing we do with the fold change, and the same thing we would do with the p-value. This we would do for each of the different factors, but and we can also configure the annotation to use if we want to get some additional information on the type of genes that are present. So in this case, we will use the UniProt annotation for human, as this data is a human data set, and we will just select a couple of annotations to apply. And then we can also configure the settings here, so we can have a look at the analysis overview. If we have information on the expression, which we have loaded already, the differential expression, which is already loaded, and we can also get some additional functional information, where we can add some summaries for the different functional categories. We can also perform a k-means clustering to identify similarities. And we can also add a custom target list that we can load from any type of, in this case we can use a gene set downloaded from the broad, but any type of gene set could be loaded here as long as it's in the GMT format. And this means that if you have a set of genes that you are particularly interested in looking at their expression, you could easily upload them into the Explorer, and it will be available during the exploration of the data. Okay, I'll just close this because I have pre-generated this analysis. So I'll cancel here, and I will close this instance of Spotfire. And here we have the results. So when you have finished executing the ExpressionExplorer, you have this initial screen where you can easily select from the different comparisons that you have loaded, those genes that are significantly different. These can be browsed in the lower part of the screen. So you can see, for example, in this case, we can see that this gene is overexpressed in several of the different comparisons. We also have the information on the fold change and the p-value. We can look at those cases that are significantly downregulated, which are marked in red. And this, you can also look from different comparisons. You have a summary of the data you've looked at. So basically you have information on the tissue, the number of samples, on the read length, and also the number of samples. So you can see that for each read length, you have one sample of each tissue. And now we can start going through the different questions that we have mentioned at the beginning. So for example, if we want to see if there's difference between technologies, we can go to the tab marked as sample expression similarity. And here we have a heat map that shows the similarity between the different groups of samples. So here you can see each of the categories, so adipose, adrenal, brain, and each of the different samples. So you have 2 samples for each. You can also look at the overall correlation by group and by each of the different groups. Or if you want, instead of looking at tissue, you can look at the read length. And in this case, you can actually see that there's not such a good similarity. So we can already see what we probably expected, that the results obtained when performing the analysis using different read links are equivalent. If we go back to the comparison by tissue, we also have information on the correlation of the different samples within the groups. So within muscle, the correlation is quite good. We can see in all these cases we have a correlation green, except in heart where we have 0.94. This is colored in red because we have set the threshold at 0.95, but it's still a quite good correlation. In addition to this heat map, we also provide a clustering. So here I'll just increase the size of this cluster so that we can see it in more detail. So here you can easily see what tissues seem more similar from the expression profile. So for example, we had adipose and breast tissue, which probably are expected to group close together. You can also see information on the expression similarity on the PCA plot. So here we can see, for example, testis and brain, which are a bit different from the rest of the groups. This is probably because there's a very high level of expression in these two tissues, which makes them quite different. But in any case, what you do see is that both samples in each of the tissue types group very closely together. So we can see that the difference due to the read length is negligible. So that way we can easily, just by looking at the plots, answer some of the questions. So we can see that there is no difference between technologies. And we can also see that the profiles are quite similar within the samples from the same tissue. If we want to see the overall expression, we can have a look at the box plots that we have here. Once again, we can look at the box plots by group or by sample. So all these visualizations are fully interactive. Another question we were asking at the beginning was if we can see the top expressed genes. So here, for example, we can select a set of tissues and view those genes that are most expressed, as well as the profile that they show. So if we want to see what profiles these genes have, we can just select the different genes. You can perform multiple selections, and then you will get the different genes present in the spark lines, and also here you will have different plots you can navigate through. Once again, as before, you can go through the sample and through the tissue, so you can see the examples group-based tissue. And another thing we can see is also the expression of the genes compared to the different samples. So for example, here you can see the expression rank of each of the genes over all the different tissues. So you can easily see which are the most expressed genes, or you can easily navigate through them and identify cases where a gene, for example, is very highly expressed in a certain tissue, but not so much in the rest, which can indicate cases where a certain gene might be more tissue specific. Okay. If we want to look at the significant differences, so here we can see the representation of the different comparisons between tissues. So this would be for the different comparisons we have loaded. We can easily see the behavior of different genes in these different comparisons. So for example, here we are selecting from the list that we loaded, which is a list downloaded from the broad, those genes involved in muscle contraction, and we can see how they behave in each of the different comparisons. I will just make it a bit larger so that you can see it better. We can select, for example, myogenesis, and this will automatically update. Or we can also go through different examples. Here we have a list of the different paths that are loaded in this custom gene set that we have added to analysis. For example, if we look at those targets that interact with nephrine, which is a gene that is usually expressed in the kidney, we can see that, for example, they seem to be more expressed actually in the kidney, which is what we expect. And if we look at the nephrine itself, we can just remove all selections and mark it on the graph. And here we can also see that it's much higher expressed in the comparison with kidney. If we go to the lower panel, here we can see this information in more detail. And we can see that actually, with the exception of the comparison of kidney versus liver, this gene is downregulated in all the different comparisons, except in the case of heart and muscle. So in this way, we could easily navigate through sets of genes that are of interest for our own experiments. So if we had loaded here a set of genes that might be involved in our disease of interest, and we had and we were looking at a set of patients, we could easily see how they are these genes are behaving in the different comparisons. So it's very easy. You can also select any combination of these comparisons that you want to look at. And for example, another of the questions that we addressed initially, is there significant differences between the different tissues, can be easily answered here, where you can, in a very easy manner, see those cases that are significantly different, those that are above the red line, and with a higher fold change, which are those that are farther away from the zero, either upregulated or downregulated in the respective tissue. So also, we can confirm what we saw before, that in most cases, there's not such a big difference in between the technologies. And if we look at the comparison in all the different tissues, except for this different read length, we can easily see that if we those genes that are upregulated in these comparisons are actually all non-significant in the comparisons between the different tissues. So we can be quite confident that there is no problem using the different region that will affect a major number of genes. Okay, so in addition to the volcano plot, you also have other options to look at the different expressions. So here we can also look at how any list of genes behave. So we automatically generate, in addition to those gene lists that have been loaded, those top genes that are expressed in each of the tissues, and those most significantly different in each of the tissue comparisons that we have provided. So we could see, for example, for the top expressed genes in a certain tissue, which is the behavior in each of the different tissues that we are looking at. Or for the top differential expression expressed genes between one of the comparisons, what is the behavior in the rest of the comparisons. Okay, so This will give us an answer to some of the other questions, such as is there significant differences between the different tissues, or which are the most expressed genes, and how do these behave in the different tissues when we look at them. The other important question that we usually like to ask is regarding functional information. So when we look at functional information, here we can also see for each of these lists of genes what are the most represented functional categories. So here, for example, I'll just make this a bit bigger in order to see it better. So here we can select for the top 500 expressed genes in white blood. We can see that, for example, most of the Go categories are related to immunities, platelet activation, innate immunity. As you can see, this is completely interactive, so you can select, whenever you select any of the different sections, you will get an update, an instantaneous update of the rest. Here we have the two Go categories that we selected in the configuration at the beginning. So here we could select any other categories. And we can get additional information by clicking on any one of them and going into the details at the bottom. Okay, so finally, another very important element that we have is one that allows us to compare the gene list that we automatically generate with those that are automatically generated by other software, or those that we have uploaded ourselves from custom gene lists. These are marked in orange. So here, for example, we can select those top 500 genes expressed in white blood, and we can see in the orange boxes that we have different categories that are related to immunity. We can also look at examples, for example, in the muscle, and we can see that we have that these are similar to those genes listed related to muscle contraction, striated muscle contraction. And for example, if we look at colon, we would also see smooth muscle contraction, which is what we would expect to see. Of course, in this case, because this data set, we know what we expect, this is not such a surprise, but if you had a set of genes that you had identified in a previous study, or several different pathways that you have identified that are altered in a different study, you can easily, just with a few clicks, run your data through this exploration tool and get all this information in a few minutes. So with this, I think I will conclude the demonstration. We could, of course, go deeper into any of the features that you would like to see in more detail, so please feel free to send any questions that you would like to address to the text box. In any case, while we are collecting these questions, I just wanted to thank you for your attention. And you can visit us at our Integromics website or at any of our Facebook, Twitter, LinkedIn, or YouTube accounts. And if you want to get more information on OmicsOffice, just feel free to visit us, or also to visit our Perkin Elmer partner that will provide you with any information you require.